Module 1. Inheritance, DNA form and function. DNA is the fundamental hereditary material of all living organisms on the Earth. In eukaryotes, DNA is stored primarily in the cell nucleus, and a small number of genes are stored in the mitochondria. Even prokaryotes, like bacteria and archaea, have DNA, but it floats around in the cytoplasm because these cells have no cellular organelles. There are 6 billion base pairs in every cell in a human body. Base pairs are about 0.34 nanometers apart, so the length of DNA in every cell is about 2 meters. If there are 2 meters of DNA in every cell, and an average person has about 75 trillion cells, then the total length of DNA in a person is about 150 trillion meters. To give you some perspective, the distance from the Earth to the Sun is about 150 billion meters. So every person has enough DNA to go from the Earth to the Sun and back 500 times. DNA is truly an amazing molecule. Its double helix structure makes it very sturdy, which explains why we can still extract DNA from ancient organisms like woolly mammoths. There are 2 copies of every DNA molecule in every cell, providing redundancy of the instructions. And the fact that we have a double helix with complementary sequence means that there's a mirror copy of the sequence in every DNA molecule. Its modular structure in a series of nucleotides makes it easily transcribable, and this nucleotide-based structure also allows for mutation and variability, which is actually really essential for the survival of a population. Its expression is regulated at many levels, allowing for high complexity within a fairly simple structure. There are 2 copies of every chromosome in diploid organisms like humans, and there is variability throughout the genome. Each form of a DNA sequence variant is called an allele. Homozygotes have 2 copies of the same allele, and heterozygotes have 2 different copies. The influence of DNA can be examined at the level of function through gene expression and protein action, or the effect of DNA sequence and variation can also be examined through segregation and inheritance. The basic elements of a gene are given here. Genes have distinct coding regions called exons that may or may not be contiguous. Between each exon are regions of intervening sequence called introns, which can be very long. The reading direction of DNA is 5' to 3', which is associated with how the molecules in the sugar backbone connect to one another. Upstream and downstream of a gene are regions of sequence associated with the gene but not part of the gene product, and these are called the 5' and 3' untranslated regions. The start of a gene is designated by the sequence ATG, and the stop sequence can be either TAA, TAG, or TGA. The sequence between the start and stop codons is called the open reading frame, or ORF. The start and stop landmarks can be used to identify potential genes within a genome, which, until verified as a gene, are often simply referred to as an ORF. Upstream of the start site is the promoter region. This region contains complementary sequence to proteins called transcription factors that you learned about when you took molecular nutrition. These enable the transcription machinery to bind to DNA and to drive the expression of genes. Enhancer and repressor sequences can either be upstream or downstream of a gene, and they can also be very far away. These sequences can physically promote or repress gene transcription. To initiate transcription, a transcription factor complex binds to DNA, enabling the transcription machinery to do its job. The initial phase of transcription replicates the gene from the start to the stop codons. A second round of processing, in which the splicing machinery removes intronic sequence, converts nuclear RNA to messenger RNA. What I just described is the classic view of how DNA sequence is converted to RNA sequence. But we now know, as you will see in a later video, that the genome contains many regulatory elements that are converted to RNA but do not encode proteins, and do influence gene expression. These molecules include long non-coding RNAs, small non-coding RNAs, and microRNAs, among others. Messenger RNA molecules are transported to the cytoplasm, where ribosomes convert RNA sequence to proteins by creating chains of amino acids based on the instructions in the RNA. The linear form of a protein is called the primary structure, but few proteins are found in this form. In 1968, the deciphering of the genetic code won Robert Hawley, Har Khorana, and Marshall Nirenberg the Nobel Prize in Physiology or Medicine for their interpretation of the genetic code and its function in protein synthesis. The genetic code is made up of codons, which are sets of 3 nucleotides that specify a given amino acid during translation. There are 64 possible combinations of 3 nucleotides, which means that for almost all amino acids, there's more than one way to encode them. Yet, this is another way in which our genome protects against deleterious mutation. The third position of a codon is called the wobble position. It is the nucleotide that can be variable in the genetic code. If a mutation occurs in the wobble position, it is less likely to cause a change in the protein than mutations that occur elsewhere. Note that the start sequence ATG encodes for methionine. As we said earlier, most proteins are not found in their primary structure. The sequence of the protein determines its secondary, tertiary, and quaternary structure. How a protein folds is critical to its function. If a mutation occurs that disturbs protein folding, the consequences can be substantial. Proteins can have many kinds of functions. They can be catalytic, or stimulate and facilitate reactions between 2 molecules. They can provide structural support for cells in the organism. They can act in the action of synthesis, where they're building or joining molecules or other parts of the cells and organism. They can cause degradation of target molecules. They can provide transport and storage of various biomolecules and metabolites. They can act in the level of signaling, and they can provide coordinated motion, as in muscle filaments or the cytoskeleton. The Human Genome Project began in 1990, and the sequencing of the entire human genome was completed in 2003. The goals of the project were to identify all genes in human DNA, determine the sequences of the 3 billion chemical base pairs that make up human DNA, to store this information in databases that could be accessible to the public, to improve tools for data analysis, to transfer any technologies developed to the private sector, to address all the ethical, legal, and social issues that were potentially expected to arise from knowing the entire sequence of the human genome, and ultimately to understand how variability in the human genome influences differences in health, behavior, and physical function. In 1991, just after the launch of the Human Genome Project, one of the leading population genetics labs in the country, led by Dr. Wensheng Li, published a seminal paper in which they estimated that the human genome contained a variable nucleotide about every 1,000 bases, for a total of about 300,000 predicted variants. But even they considered this estimate to be low and suggested 3 possible explanations: that the mutation rate in humans is low, that the effective population size of the human species has been relatively small, or that the species has undergone a severe bottleneck. These are population genetics terms that you don't really need to know, but they refer to the hypothesis that the human population was fairly limited in its growth, and that would limit the spread of mutation within the population. But the real problem was that they based these assumptions on 75,000 bases of sequence that they had generated in their lab. The technology at the time involved running separate sequencing reactions for T, A, G, and C, in which each nucleotide was labeled with a radioactive tag, followed by loading these reaction products onto a gel medium and exposing the reactions to an electrical current to separate molecules of different sizes. The gel was then exposed to a radiographic film, and a lab tech, or more likely a lowly grad student, then literally took a ruler, moved it down the film one row at a time, and read bases out loud while a colleague wrote them down. You can imagine that to generate 75,000 bases in this manner was pretty extraordinary at the time. And to give you some perspective, this work took many years, and it is now possible to sequence an entire 6 billion nucleotide human genome in about a week. As of May 2000, when the first drafts of the human genome were being generated, a little over 135,000 variants, or single nucleotide polymorphisms, or SNPs as they're known, had been identified and cataloged in the NCBI dbSNP database. By January 2002, a total of 4 million SNPs had been entered in dbSNP, and as of April 21, 2011, a total of 41 million SNPs had been identified. The latest statistics from dbSNP indicate that there are more than 113 million validated SNPs currently in the database. Although this seems like a lot of variation, keep in mind that it's actually less than 2% of the entire human genome. When you look around you at the variety of shapes, faces, sizes, and colors in the human population, it's important to realize that we are all essentially the same.